My screen is visible. Hmm. Okay. 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 So we start today's lecture. In last lecture, we will discuss about the sequence retrieval and structural analysis. In today's lecture, we will discuss about the T cells epitope prediction and V cells epitope prediction. Okay. So for for predicting the T cells and V cells, we use a software that is the IEDB. Okay. For IEDB, in Google, we simply search IEDB. IEDB database. This is the IEDB for free epitope database for the production resources. Then open this first link. Now this is the home page. Linear, peptide, discontinuous, non-peptide. This is the home page. There are lots of options are available. We use these options according to our requirement. Okay. For predicting the epitopes, we simply click on analysis, then T cells epitopes. Okay. First we isolate the peptide binding of MHC class 1 molecule. Okay, this one will open. Then paste the sequences in FASTA format. Okay, and for my sequences, wait a minute, I will show you the sequences. Mm, mm, mm. Give me a minute, I will show you this is the result. Mm. And this is your sequences. Okay, now I copy the sequences in FASTA format, then paste. Okay, and we also upload the file from here. Okay, here this is the method by which we predict the epitopes. For this we simply search ANN 4.0. Okay, okay, then we select the MHC molecules. For selecting the MHC molecule, you simply click on this question mark, and this is the method for epitopes. Then again move, then click on this selected HLA allele reference sets. Okay, whenever you click on the reference set, then all the alleles are selected. Okay, then submit and wait for the result. Okay, again move to analysis, then T cells, then B cells epitopes. For MSc class, second epitopes. In again, paste the sequence in FASTA format. Here for predicting method is this one. Is this one. Okay. Then again click on this question mark. This is the reference of allele. Then click on this references sets. Now all references are selected. Okay. Then select the default value. Default value is 15. Then submit and wait for the result. Now this is the epitopes of MSc class first in your giving sequences. Understood? Or this is the result of MSc class second. MSc class second or MSc class first. These are the epitopes. These are the alleles. This is the starting point, end point, length, peptide, IC value and rank. Same for here. Okay? Clear? 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 Okay, Anjali. Abdul, Afreen, Angel, Chandan, Bhargavi, Bala, Shri Lakshmi. Okay. Moni, Suma, Gayatri, Malar. Okay. Now, again, this is the MSc class second, MSc class first. Now for the B cells epitopes. For the B cells epitopes, click, then click predicting the linear epitopes, then paste the sequences in only protein sequences. So I remove this FASTA format. Okay, and this is I have selected these options and submit and wait for the result. Okay. Understood? All clear? Follow. Tell me now. This is the result of B cells epitopes. Okay. The threshold volume means more than, for B cells epitopes, the threshold value is always higher than 4. Center position is 4. The threshold value is 0.550. Okay. And this is the average value. This is the maximum and this is maximum and this is the minimum value. And average means 0.4. More the 4 value, more the appropriate results. Okay. This is the number of epitopes. Number of epitopes is 5. This is the starting point. This is the end point. This is the sequences and this is the length. Or in this residue table, These are the amino acids which are participate in a formation of epitopes. E denote the epitopes. Understood? This one. This one. This one. This one. This one. Okay. Now this is the result of B cells epitopes, T cells epitope, MSC class 2 and MSC class 5. Now tell me, clear? जल्दी बताओ. श्री लक्ष्मी, प्रशांत.
Yes, ma'am.
क्लियर श्री लक्ष्मी?
Yes, ma'am.
कोई डाउट? प्रशांत.
इंटरप्रिटेशन को दोबारा बताइएगा एक बार.
इंटरप्रिटेशन देखो बी सेल्स एपिटोप्स की, देखो ये है आपकी एवरेज वैल्यू की. एवरेज वैल्यू आपकी 0.4 होनी चाहिए. ओके. मिनिमम वैल्यू आपकी 0.24 जा सकती है, मैक्सिमम 0.88. तो बी सेल्स एपिटोप्स होने के लिए जो उसकी थ्रेशोल्ड वैल्यू है वो 0.4 से अबव होनी चाहिए. तो जिस रीजन की थ्रेशोल्ड वैल्यू और स्कोरिंग वैल्यू 0.4 से ज़्यादा होगी वो हमारा एपिटोप की कंडीशन में आएगा. और कौन से एपिटोप की श्रेणी में आएगा? बी सेल्स एपिटोप्स के पोजीशन पे आएगा. तो देखो ये सारी आपकी पेप्टाइड लेंथ है, ये आपके सारी लेंथ है और उनकी थ्रेशोल्ड वैल्यू इसमें गिविंग नहीं है, लेकिन सबकी थ्रेशोल्ड वैल्यू के लिए हम ग्राफ देख सकते हैं. ये सारी वैल्यू 0.4 से बड़ी ज़्यादा है, इसीलिए ये हमारी इस पर्टिकुलर प्रोटीन में टोटल 6 बी सेल्स एपिटोप्स प्रेजेंट है. नाउ क्लियर?
टोटल 6 में कहाँ पे लिखा हुआ है? यहाँ पे
क्या चाहिए रहा? और देखो थ्रेशोल्ड वैल्यू ये भी दे रखी है.
ये सिर्फ बी सेल का है ये.
हाँ, इसे बी सेल्स के लिए है.
ओके.
समझ में आया? अब आ जाओ टी सेल्स पे. टी सेल्स पे भी आपकी जो वैल्यूज होती हैं ये वाली. ये सारी स्कोरिंग वैल्यूज लिख के आती है इसमें. पहले इसको हम कॉपी करते हैं, फिर पेस्ट करते हैं, अपनी एक्सेल फाइल खोलते हैं, ओपन आवर एक्सेल फाइल. ओके एंड पेस्ट. अब देखो सब एक ही में आया रिजल्ट. ये देखो ये आ गया आपका रिजल्ट और ये क्लियरली विजुलाइज है. देखिए ये वैल्यूज आई हैं और ये सारी वैल्यू आपकी 0.4 से अगर एबव होगी तो वो पर्टिकुलर रीजन आपका एपिटोप्स के लिए होगा. ओके ओके Okay, प्रशांत
Yes, ma'am.
समझ आया?
Yes, ma'am.
अब एमएससी क्लास फर्स्ट के लिए भी सेम करो, कॉपी करो. कंट्रोल ए, कंट्रोल सी एंड कंट्रोल बोलो कंट्रोल, बोलो क्या हो गया? समझ में नहीं आ रहा है क्या? बताओ. पेस्ट करने के लिए क्या करना पड़ेगा?
कंट्रोल पी.
कंट्रोल पी,
कंट्रोल वी.
कंट्रोल वी. और ये क्या रिजल्ट हमें आई है? ये रिजल्ट हमारा किस चीज का है? ये सारे रिजल्ट हमें किस चीज के आए हैं? बताओ पहले मुझे, फिर आगे बढ़ेंगे.
T select.
किसके रिजल्ट है ये? These are the result of. These are the
T cell.
results. These are the result of. These are the result of. Prashant.
Yes, ma'am.
These are the results of.
T cell, I think.
I think T cells.
MHC molecules result. The result of MHC molecules class 1 and 2.
हाँ. These are Chandan, Gayatri. Bhargavi. These are the results of. Mufida, these are the results of. Mufida, these are the results of. हाँ Abdul, you are the right, but other one.
MHC finding
MHC class 1, class 2 molecule from T cells, and these are the results of epitopes.
T cell epitope.
T cells epitopes and B cells epitopes. Prashant, this is the result of.
This one is the result of one only, no? Means
This is the result of B cells epitopes or T cells epitopes.
वो मिक्स हो गया, पता नहीं.
सब मिक्स हो गया सबका. मुफिदा, श्री लक्ष्मी, राहुल, प्रज्ञान, what T cells? देखो, this is the result of B cells epitopes. This is the result of T cells epitopes, but MSC class second molecule. And this is the result of MSC class first molecule or epitopes. T cells epitopes and B cells epitopes. Understood? Mufida, Pragyan, Prashant, Rahul. Now clear?
Okay, ma'am.
Now tell me, this is the result of
Cell E.
T cells.
E cell cell.
T cells epitope. MSc class 1, MSc class 2.
Class
MSc class 1 or MSc class 2?
2, ma'am.
2. Or this is the result of
E cell
B cells epitope. And this value, this yellow line indicate
above 0.495 epitope.
these yellow color denotes the number of epitopes, and this green color is for yellow is for the epitopes, and the green is for the non-epitopes. Okay, and this is your final result. This is the result of MSc class 1 molecule, and this is the result of MSc class 2 molecule. Okay. Okay. Tell me yes or no.
Yes, ma'am.
Okay. Now we filter out these epitopes. Okay. For filtering these epitopes, we simply go to the data, then filter. Selects the peptides, filter. Now filter will apply. Okay. These are the different molecules, or these are the total number of peptide chain. Okay. Now I select this first one. Only simply apply the filter, then select only first one, click. Now this is only a peptide chain for all the epitopes. Now for checking the allergicity of this molecule, which tool we use? For checking the epitope, checking the allergenic nature, which software we use? एलर्जेनिक नेचर को चेक करने के लिए विच सॉफ्टवेयर वी यूज? Allotop. Okay, so now we check the allergenicity and antigenicity. For checking the antigenicity, which software we use? So in Google, in simple, we simply search Vaxizine and Allertop. Okay, open this one. Wait a minute. Okay. Okay, now paste the sequences, and your sequences is this one. So simply copy the sequences, copy the molecule, Okay. Hello. Hello. Hello.
Yes, ma'am.
Paste and submit. Now your molecule is allergenic or non-allergenic?
Allergen.
Allergenic. Check the antigenicity. Antigenic or non-antigenic? Antigenic or non-antigenic?
Antigenic.
Antigenic. But this molecule have a antigenic property but have a allergen. That's why we does not select this. Okay. Now check second one. For checking the second one, you simply click on filter, deselect this first one, select first one, second one, and OK. Then copy the sequences, and paste. I think this compound is allergenic or antigenicity or non-antigen?
Antigenic.
For allergicity, paste and submit. Non-allergen.
Non-allergen.
Means we select this epitopes or not?
Yes, select.
Yes. तो देखो इसी तरीके से हम फर्स्ट ईच एंड एवरी मॉलिक्यूल की एंटीजेनिसिटी चेक करेंगे, एलर्जिसिटी चेक करेंगे. अगर वो हमारा नॉन एलर्जेनिक है तब भी हम सिलेक्ट करेंगे, नॉन पैथोजन है तभी हम सिलेक्ट करेंगे फॉर द वैक्सीन पर्पस. अंडरस्टूड?
नॉन एंटीजेनिक है तो
एंटीजेनिक और नॉन एलर्जेनिक. अरे देखो नॉन एलर्जेनिक का मतलब क्या हुआ, एलर्जन का मतलब व्हाट इज एलर्जेंस? कैन यू प्लीज एक्सप्लेन मी व्हाट आर द एंटीजेनिसिटी एंड व्हाट इज द एलर्जन?
any foreign substance that elicit immune response, like possession of the T-cells that go to this antibody against disease. Allergen are not.
तो फिर ये होगा आपके एंटीजेनिसिटी, मीन्स कोई डिजीज कॉज करे, एलर्जेनिसिटी कोई एलर्जी कॉज करे इन ह्यूमन बॉडी. ओके, ये मॉलिक्यूल सेकंड वन हमारा नॉन एलर्जेनिक है और नॉन एंटीजेनिसिटी है. ओके, एज सेम we check the allergicity or antigenicity of each residues. Understood? Now same for the B cells epitopes or B cells epitopes. Check okay. Now for MSc class second, how we apply the filter? How we apply the filter?
Data.
Data.
Their data.
Database. Right.
Just to go to data.
Go to data.
Data and
Check the
apply.
allergicity. Check the allergicity or antigenicity. Data and
Data and apply filter.
filter. Now filter will apply. Now check the antigenicity or allergicity of each molecule. These are the number of epitopes. Understood? Deselect and select this first one and apply. Now tell me how we check the allergenicity or antigenicity of this molecule. How? Tell me. बोलो. Tell me.
तो कॉपी करके जो ये यहाँ पे आप
Copy.
और उसके बाद एलर टॉप पे जाकर वहाँ इसको पेस्ट करें
And this software is used for
एलर्जेनिक
allergenicity. Your molecule is
Allergenic.
non-allergenic. This molecule is allergenic. Or antigenicity or non-antigenicity. And submit. Antigen or non-antigen? Antigen or non-antigen?
Antigen.
Antigen.
Means allergen or antigen. Can we select this one? Allergenic as well as antigenicity. Can we select?
No.
No. Okay, like this. For one by one we select and check the antigenicity or allergenicity. Okay, after checking the antigenicity and allergenicity, we construct the vaccine.
for each area.
Okay, then simply second one, first one. Okay, again check the antigenicity or allergicity. After checking the antigenicity or allergicity, we find this result. Okay, this is the number of amino acid which are participate and this is the amino acid sequences from MHC class 1 molecule, or these are the epitopes from MHC class 1 molecule. On niche, okay, and this is the MHC class 2 molecules.
And for each molecule we check color.
बोलो.
For each molecule I will check color.
How? Which molecule?
Every not the every
All molecule. समझ नहीं आया क्या बोला?
सारे molecules को check कर.
हाँ. Check each and every epitopes. Okay. After checking the epitopes, you will make a Excel sheet like this. These are the MSC class 1 molecule. These are the MSC class 2 molecule, and these are the linker. Introduce. Then this is the ID. Kiel grasshopper. Or the a linker. MSC class 1 molecule, MSC class 2 molecule, linker, and this is the histidine molecules. 6 histidine residues. Okay. Then first we add the linker, then B cells epitopes, then again linker, then MSC class 1 epitopes, then again linker and MSC class 2 residues, then histidine tags, and this is the final molecules finally the sequences of our vaccine. Understood? Hmm? Hmm?
Yes, ma'am.
देखो, first we select the apply the filter. MSC class second residue. Check the antigenicity and allergenicity. After checking the antigenicity and allergenicity, we note down into the another Excel sheet. Okay. After filtering out, we find this one result. This result. Okay, these are the in B cells epitopes we does not check any antigenicity or allergenicity, we directly use. But for the MSC class 1 molecule or MSC class 2 molecule, we check the antigenicity as well as allergenicity. Then we find this linker. Okay, and for linker, finding the linker, there is another database is available, that is the linker database. Linker database. Okay, then open this database, then search your protein like membrane protein and search. Now this is your result. Reasons, chain, description. Okay, and for checking the linker, we also use the literature. Okay, first we read the literature, then by the help of this literature we find the linkers. Understood? Okay, and this is our vaccine structure. Okay, yes. Okay, Anjali. Okay, this is the MSC class 1. This is the complete constructions. Dekho. First we check the epitopes. For epitopes we use IE-DV molecules. For checking the vaccine, for checking the antigenicity or allergenicity, we use Vaccegen and Allotop. Okay, then we construct the vaccine. For constructing the vaccine, first linker, then B cells epitope, then again linker, then T cells MSC class 1 molecule epitopes, then again linker, then MSC class 2 epitopes, then histidine molecule. Or histidine number of histidine molecule is 6. Okay. Now our vaccine is completely construct. Clear? Any doubt? Any question? Okay.
Is histidine molecules the 6 for all vaccines, or is it different? Like for bacterial everything, it will be different or same? You have given 6 histidine molecules.
6 histidine value. Sometime it will vary, but sometime it also conserve.
Okay. So how many we have to include? How you will know?
For literature. For this we go to the literatures.
Okay.
Okay. Understood? And Ambivati, I think you are absent in last lecture, no?
Yeah. I have it.
And okay. You have any information? You have any idea about the vaccine designing?
No, ma'am. Actually, I'm a consultant microbiologist. By seeing regarding this workshop, I was interested.
Okay, but how you understood today's topic?
Yeah, I understood. I'm a microbiologist.
Okay.
Medical microbiologist.
Medical microbiologist. Okay. B cells epitopes, C cells epitopes, antigenicity, allergenicity, and construction is clear?
Yes.
Okay. Now we use this option, that is the vaccine expression analysis. For vaccine expression analysis, We will discuss this docking and analysis in next section, but in today's session we will discuss about this vaccine expression and analysis. Okay, for checking the vaccine expression and analysis, first we convert this mRNA sequences or this protein sequences into the nucleotide sequences. For this we use a software that is known as a Bactrans. Use Bactrans. This is the embossed Bactrans sequences. By the help of this software, we change the protein sequences into the nucleotide sequences. Protein sequences convert into the nucleotide sequences. Okay. Copy the sequences and paste. Okay, and we also upload the file, then parameters, then name, and submit. Okay, wait for a minute. Now this is the protein sequences or nucleotide sequences.
Nucleotide.
Sequences. Matlab we put the protein sequences, and protein sequences means we put the vaccine sequences. Okay, then we obtain the nucleotide sequences from this by the help of this software. Okay, then we optimize this nucleotide sequences. For optimizing, we use a software that is the ZCAP codon optimization. Okay. Then open this thing, that is the JCAT. Okay, advance. And okay. And paste the sequences, and this is our sequences. So I copy sequences and paste and select the check. Okay, and select the restriction site. Let's suppose I select this one. Okay, then click on submit. Okay, wait a minute. Okay, wait. I think there are some issues. I does not select. First we select the organisms. It is the Homo sapiens, and submit. Now this is your result. This is your pasted sequences, and this is your optimized sequences. Okay. Their CT value, their number will increases. Okay. Now we use this for the further analysis. I simply copy these sequences and paste here. And remove these. Okay, now this is your improved sequence. Understood how we optimize our sequences? Understood? Clear? Okay. Okay, Gayatri. Sri Lakshmi,
Yes, ma'am.
clear?
Yes, ma'am.
Gayatri,
Yes, ma'am.
clear?
Yes, ma'am.
Okay. Okay, Pragyan. Okay. देखो वैक्सीन एक्सप्रेशन फर्स्ट स्टेप कंप्लीट सेकंड स्टेप नेक्स्ट एम्पलीफिकेशन और क्लोनिंग नाउ वी डिस्कस दिस पार्ट क्लोनिंग इज वेरी इंपॉर्टेंट ना फर्स्ट यू प्लीज टेल मी व्हाट इज द क्लोनिंग what is cloning
Cloning is a procedure in which we create multiple copies of a same gene or an organism.
and what is the actual procedure of cloning
In cloning, when we apply genetic engineering principles for our RDT processes,
what is the what principle
ma'am, recombinant DNA technology or genetic engineering के principles.
actual process key steps what are the steps like cloning restriction and endonuclease like this what are the steps of cloning
Ma'am, first of all, we will select a gene of interest from a particular DNA, then treat them with appropriate endonuclease enzyme. Then after that, we will link
which
it with
enzyme
the
endonuclease which
restriction
one?
endonucleases.
What is the restriction endonuclease enzyme
Ma'am, restriction endonuclease is a kind of nucleases enzyme which is meant for cutting DNA at a specific site.
then
Then after that, ma'am, we will take into account the host cell DNA and we will ligate the DNA that we have been cut
for
from
ligation
the
process for ligation
ma'am,
process
ligation लिंकिंग प्रोसेस में जिसमें ब्लड उसके जो स्टिकी एंड्स होंगे उनको हम लिंक कर देंगे आपस में
for ligation process we use the DNA ligase enzyme after the ligation process
From after ligation process they are transferred to a cloning vector.
Madhuri, please mute yourself. देखो what is the cloning process in cloning process first we take the vector vector first we take the vector and gene of interest then cut this gene of interest and vector with restriction endonuclease enzyme then by the help of ligation DNA ligase we ligate the enzyme then transform into the host cell this is the cloning process okay and during the vaccine this vector and restriction endonuclease enzyme also plays a most important role okay for the cloning purpose we use a software like BenchLink like SnapGene this SnapGene is the paid software and this BenchLink is a free software okay what is the actual BenchLink what is the actual working of BenchLink we will discuss in next session okay and today's session vaccine construction is clear
Okay. Yes, ma'am.
clear vaccine construction
Yes, ma'am.
okay means step number first clear step number first clear
Yes.
step number second epitope prediction. Step number 3.
Yes. Yes, ma'am.
Now in next session we will discuss about this fourth secondary tertiary structure we already discussed. Okay, molecular dynamics and expression analysis. We did in next lecture we will discuss about the benchling and docking softwares and simulation. Okay, clear?
Yes, ma'am.
Okay, then thank you, class.
Ma'am, where can we find the recording of previous session and this session, ma'am?
Sir will send
Okay.
on your portal.
Okay. I did not receive yesterday's also. Once I want to do it.
Okay, I will discuss this with sir. Okay.
Okay. Right.
Okay.
Thank you.
Okay, welcome. Thank you, class.
Thank you.
Thank you, ma'am.
Welcome.
Thank you, ma'am.